Hi, welcome. Let the day fall away now. There's something beautiful about the idea that we are written in code, each of us, a long sentence in an ancient language. And now, for the first time, that sentence can be rewritten by our own hands, they say. 2 girls were born this way, edited, designed, not loudly, just quietly, like a ripple in still water. Where are you as you listen to this quiet question? You lie still, eyes heavy, breath soft, as night gathers gently around you. Somewhere in the distance, the world hums its lullaby, a cricket's sigh, a breeze across leaves, the quiet pulse of time. And deep within you, far quieter still, a story begins to whisper. It lives not in your dreams, but in your very cells. A secret symphony, older than memory, older than language, older than the stars that watched your ancestors walk beneath them. You are made of light and dust, yes, but also of code. Not the kind typed on a keyboard or stored on a screen, but something deeper, older, a kind of biological poetry. Every one of your trillions of cells holds a complete copy of it, curled tightly, safely into threads, so fine they are invisible. This is your DNA, your deoxyribonucleic acid. But we won't use long names tonight. Let's stay simple. Let's say this. You are made of letters. 4 letters, repeating in patterns so intricate and wise, they have built every part of you. A for adenine, T for thymine, C for cytosine, T for guanine. They are not just letters. Of course they are molecules shaped to fit each other like lovers' hands. A always pairs with TC, with G, a secret handshake passed down through generations. Now imagine a ladder, a spiral staircase, so fine it can barely be seen. This is your DNA, the rungs of this ladder are made of those pairs, A with T, C with G, a dance of opposites. The sides of the ladder are sugar and phosphate, holding everything in place, twisting upwards in a double helix, like vines climbing toward the sun. This spiral lives in the heart of your cells, folded again and again until it becomes compact, enough to fit, yet still vast enough to hold the entire recipe for you. This is your genome, your biological autobiography. Every strand of your DNA is like a line in a long book. What a book it is! 3 billion letters long. If you were to read it aloud, 1 letter per second, day and night, never stopping, it would take you nearly a century. And yet, your cells read parts of it every moment of your life. They don't read it front to back like we do. They read pieces as needed, as though flipping to just the right page, just the right sentence, whenever a new protein must be made, or a wound must be healed, or a memory must be formed. It's a modular language, flexible, responsive, and beautifully precise. These sequences of letters form genes. Think of genes as instructions. Some are short, a few hundred letters long. Others sprawl across thousands. Each gene is like a recipe, a step-by-step guide for building something. A protein, a pigment, an enzyme, a signal. These are the workers, the tools, the messengers of your body. They carry out nearly everything that happens within you. One gene might shape the curl of your hair. Another might influence the rhythm of your heart. Another still how you taste bitterness or see in low light. It is not a single story, but thousands layered, overlapping, whispering to each other through chemical signals. It is as though your body is a vast orchestra and your genome the sheet music. But there is something more mysterious, more profound, because this code is not just yours. It is shared. It carries echoes of those who came before. Your mother, your father, their parents, and theirs. Stretch far enough back, and you carry the code of ancient fish, single-celled swimmers in primordial oceans, of creatures who once breathed water instead of air. You are, in a way, a living fossil. The code inside you is not new. It has simply been revised, copied, sometimes miscopied, over billions of years. A mutation here, a rearrangement there, and slowly the story of life unfolds. And yet, despite this ancient heritage, your DNA is uniquely yours. Unless you have an identical twin, no one else has the exact same sequence. Even then, as life happens as you breathe, age, grow, heal, tiny changes creep in. Little edits. A molecular fingerprint left by your days. Your DNA becomes a diary, not just of your ancestry, but of your journey. So how does this code know what to do? It begins with transcription. The DNA snug inside the nucleus of your cell unzips a small portion of its spiral. A molecule called RNA copies a gene, like a scribe taking notes from a holy text. This RNA strand then floats out into the cell, carrying its message. It finds a ribosome, a tiny molecular machine, and there the words are translated, not into sentences, but into proteins. The shapes that form muscles, the enzymes that spark reactions, the hormones that carry feelings, from letters to words to function. It is biology's quiet machinery, turning instruction into existence. And yet, it all feels so still inside you now, as you lie here, cocooned in rest. Your cells are at work, faithfully, reading, building, repairing. Every moment, tiny miracles unfold unnoticed. The lining of your stomach regenerates. Neurons fire in silent symphony. Your skin renews. Your blood flows, guided by proteins made from a script 3 billion letters long. This is not just chemistry. It is choreography. And the most astonishing part, all of this began in a single cell. When you were no more than a flicker, a fertilized egg, you already carried this full code. Every cell that followed was copied from that one, carrying the same precious spiral. Through division after division, the script remained and specialized, like a play being performed by many actors in many roles. Bone cells, brain cells, skin cells, all different, yet all reading from the same script, just different lines. And somehow, from this, you became you. So tonight, let yourself marvel, not in a loud, analytical way, but quietly, in awe, in wonder, in gratitude for this ancient book that writes itself anew each day. You do not have to understand every detail to feel its presence. Just know, inside you is a language that life has spoken for 4 billion years, and it is still speaking now, in every heartbeat, in every breath, And perhaps now you feel a little closer to the mystery of being. But still, a question lingers gently at the edge of sleep. If this code shapes us so completely, who or what holds the pen? The night deepens, and you drift further inward, gently floating in that space between wakefulness and dreaming. Your breath rises and falls like ocean tides, slow and rhythmic. Around you, the world softens, and within you, the code continues to hum, the double helix, spiraling through your cells like an endless lullaby. But tonight, you follow something new. Imagine, deep inside a single cell, a traveler moving along the winding staircases of DNA. It is small, unimaginably small, yet it carries great purpose. It is not alive in the way you are, but it acts with delicate precision. This traveler is a tool, a molecular instrument, a pair of scissors designed to cut the very threads of your genetic story. And its name is CRISPR, a soft name, though its meaning is sharp. CRISPR, clustered regularly interspaced, short palindromic repeats. But don't worry about the full name. Just hold on to the feeling of it, a pattern, a rhythm, a memory coded into the DNA of ancient bacteria. Because that is where CRISPR was first found, not in humans, but in microbes. Small, simple beings. But even they, like us, have stories. And their stories, too, have enemies. In the oceans, in the soil, in the air, viruses swirl. Not the kind that make you sick, but bacterial viruses, phages, tiny hunters, seeking cells. To invade their only purpose, to copy and spread. When one infects a bacterium, it injects its own DNA, rewriting the host's code, hijacking its life. But the bacteria have learned. Over billions of years, they've evolved a memory of these invaders. When a bacterium survives an attack, it snips out a small piece of the virus's DNA and stores it inside its own genome, like a photograph tucked into an album. These viral snapshots line up in repeating patterns, interspaced, clustered, regular, short, palindromic, CRISPR. Each one is a fragment of a past war, a record of every foe that tried to invade. And when the same virus returns, the bacterium knows it. It reads the memory. It arms itself with a guide, a piece of RNA shaped from the CRISPR memory, and pairs it with a sharp companion called Cas9. Cas9 is the blade. Together, they search. They float through the cell, through the loops and spirals of DNA, until they find a perfect match, a stretch of code that looks just like the old invader. And when they do, Cas9 cuts, cleanly, quietly, a single snip, and the virus is silenced. This is how CRISPR began, as immune memory, as defense, as survival. But now, humans have borrowed this ancient tool. We have learned to guide it, not against viruses, but into our own DNA. And here, the story opens wider. In labs across the world, under soft lights and quiet hums of equipment, scientists work with CRISPR. They do not wield it like surgeons or sculptors, but more like gardeners, with care, with vision, then understanding that life is delicate and powerful. CRISPR, can do what once seemed impossible. It can find a single word in your 3 billion letter code and cut it, or change it, or replace it with something new. Imagine searching through a vast forest of letters for one tiny tree that leans slightly left. That is what CRISPR does. It uses a guide, a piece of RNA shaped like a whisper, coded to find a precise place in the genome. And when it gets there, Cas9 follows. It cuts both strands of the DNA, a clean break, and what happens after the cut is the most delicate part of all, because your cells are always ready to repair. They do not like broken strands. They try to heal. Sometimes they seal the break quickly, carelessly introducing tiny errors. A gene may be disabled, switched off, other times with help. The cell can use a template, a new version of the code, and stitch it in place, like patching fabric with finer thread. In this way, mutations can be corrected, diseases undone, traits rewritten, all with a pair of molecular scissors floating silently inside the body. It feels magical, doesn't it? Like editing life with a feather's touch. But it is not magic. It is chemistry, precision, timing, a deep understanding of the body's machinery. And yet, it still carries the weight of wonder. With CRISPR, begin to see the possibility of curing diseases that once defined lifetimes. Sickle cell anemia, cystic fibrosis, muscular dystrophy, genetic blindness. Conditions written into the DNA now, perhaps erasable, editable, treatable, not with drugs, but with code. There is poetry in that, not because it is perfect, not yet, but because it offers hope, a different kind of healing. Still, we must move slowly, for each change we make in the genome ripples outward. The cut is precise, but life is connected. Every gene touches others. Every system is a web. And in this web, a small shift can echo in unexpected ways. That is why scientists test and retest in cells, in animals, in models of increasing complexity. Each experiment brings us closer to clarity, but also reminds us of how much we still do not know. CRISPR is not a cure-all, not a magic wand. It is a key, and a key is only as powerful as the care with which we use it. Some dream of editing embryos, shaping life before it begins. Others focus on somatic cells, treating the sick without altering the line of inheritance. These are different paths, different ethics, different implications. And you, lying here tonight, don't need to decide which future is right, but you can imagine them. You can float above the present and peer gently into the tomorrow that CRISPR invites. Perhaps in the near future, a child with a rare blood disorder receives an infusion and their own cells, edited with CRISPR, begin to heal them from within. Perhaps a farmer grows crops that resist drought, not through chemicals, but through quiet genetic edits, born of CRISPR's touch. Perhaps even cancer, that tangled, defiant growth, is someday unraveled, gene by gene, until it can no longer hide from the body's defenses. CRISPR is not just a tool for science. It is a story of listening to bacteria, to biology, to the language of life itself. It is a reminder that even the smallest beings, facing their own Survival may carry answers for us all, and still, questions remain. What should we change? And what should we leave untouched? Where does healing end and enhancement begin? How do we use these scissors with grace, with restraint, with wisdom? These are not easy questions, but they are beautiful ones, because they ask us to look inward, not just into cells, but into ourselves. And as you drift towards sleep, the image lingers, a tiny pair of scissors, floating through a cell, searching for a sentence to mend. But if we can cut and repair the script of life, can we also learn to write something entirely new? You drift deeper into the quiet. The world beyond your eyelids softens, then disappears. In this stillness, there is a hum, not loud, not sharp, but constant. A gentle vibration, like the soft current of a river moving far beneath the surface. You are floating now, inside thought, inside time, and just ahead, something waits. A single cell, suspended in fluid and light. It is smaller than a speck of dust, yet its weight is immeasurable. It does not think, it does not feel, but it contains the blueprints for a human life. It holds in a soft spiral of molecules a potential future, one that has not yet taken shape, but soon will. Here, in a quiet lab, a whisper has begun, a whisper written in molecules in motion, a whisper shaped by CRISPR. This cell is an embryo, the earliest form of life, no larger than a pinprick, no older than a breath, and yet it already carries the entire library of its future. Eye color, blood type, height, the curve of a smile, the strength of the heart, a story waiting to unfold, gene by gene. But now, imagine if we could pause this story just long enough to revise a word, a mutation corrected, a disorder prevented, perhaps even a trait enhanced, vision made sharper, or memory deeper. The tool is the same. CRISPR, the gentle scissors you've already met, but the timing is different now. This cut comes before the story begins, not in an adult, not in a child, but in a single fertilized egg, in that moment before cells divide, before the body forms, before the world takes notice, and in that pause, possibility blooms. Scientists call this germline editing, a change not just for one person, but for every future child that person might one day have, because the edits made here ripple forward. They echo. They pass from cell to cell, from generation to generation. They don't just heal, they shape. So you wonder, what could we change? And should we? Perhaps it starts with something gentle, a rare disease, a life-limiting mutation. Child is spared from suffering, a family spared from loss. In these moments, the edit feels like mercy, a gift, a light in the dark. But science does not stop with what is possible. It steps softly toward what could be. What if we could reduce the risk of cancer before birth? What if we could strengthen immunity, or extend life, or soften the edges of aging? And if health can be shaped, what about other traits? Here, the lines begin to blur between therapy and enhancement, between compassion and control. Would you choose to make your child taller, or more athletic, or more intelligent? Would you remove the possibility of melancholy, or doubt, or forgetfulness? Would you, in your effort to give them the best life, edit out the very things that make life complex and beautiful and human? You float now in the space between science and story, between what can be done and what should be done in some corners of the world. Researchers have already stepped into this space. In 2018, a scientist announced that he had edited embryos, not to cure disease, but to make them resistant to a virus. There are succeeding to it. The grade. Scientists condemned. Governments recoiled, because even the gentlest cut, when made too soon, can leave deep scars. We are just beginning to understand the language of life. We do not yet speak it fluently. A single gene may do many things. An edit meant to help may have hidden costs, and the choices we make now may echo for lifetimes we'll never see. So we pause, and yet we imagine. You may picture a future clinic, clean and bright, where parents sit with counselors and scientists, looking not at a child, but at possibilities. A string of letters on a screen, a chart of genes, a menu of edits. You may see hope. In this vision, you may see danger. Most likely, you see both. For what is more tender than a child not yet born? What is more powerful than shaping a life before it begins? This is the crossroads where CRISPR now stands, in the heart of hope, in the edge of ethics. It invites us to look inward, not only at what we want for the future, but at who we are in the present. And as you float in thought, let yourself consider, do we shape the unborn to spare them suffering, or to fulfill our own ideas of perfection? Will this power unite us, or divide us more deeply between those who can afford such edits and those who cannot? Will we celebrate difference, or begin to erase it? You do not need answers tonight, only the courage to wonder. The embryo still floats in your mind, a cell so small, yet filled with such light. And in the quiet hum of the lab, you sense something more than technology. You sense longing, hope, uncertainty, reverence, because this work is not about controlling life at its best. It is about honoring it, listening closely, editing gently, remembering always that what we touch, we must touch with care. For even the smallest change in the beginning can shape the whole journey ahead. And as you exhale, slow and soft, you feel the future stretch before you, not as a single path, but as a web, delicate, infinite, waiting. Perhaps the power to edit life is not only a question of science, but also a question of story. And so we wonder, as sleep draws near, if we can rewrite our beginning. Might we also learn how to choose a new kind of end? The night is hushed, as if holding its breath. You feel it, the delicate weight of silence, a softness that wraps around your thoughts. In this space, history doesn't feel distant. It feels like it's still unfolding, like a breath caught in your chest. Tonight, we remember a moment when the future arrived before we were ready. It began quietly with an experiment, a single lab, a single scientist, a single act that crossed the threshold the world had only imagined. In late 2018, in a hospital in southern China, 2 baby girls were born. They entered the world with dark eyes, tiny fingers, and the same soft breath all newborns take. But inside their cells, something had changed. They carried edits, tiny, targeted edits made not by nature, but by design. The tool was CRISPR, the same molecular scissors you've already followed through spirals of DNA. But this time, the cuts came before birth. This time, He Jiankui, a name that now floats through the annals of scientific controversy, like a stone dropped into still water, the ripples still spreading. He wasn't trying to cure a deadly disease already present. He was trying to protect the babies from a virus, HIV. He wanted to disable a gene called CCR5, which acts like a doorway for the virus. Close the door, he thought, and maybe they'll never get sick. It was a bold idea. Perhaps somewhere in it there was compassion, but it was also premature, risky, deeply unproven. And it wasn't just about science. It was about permission, about consent, about whether the human genome. Conversation. He Jiankui moved in secrecy. He bypassed oversight. He made the edit quietly, then waited for birth. When he announced the twins, the world blinked. Then, but because someone had already used it this way, before regulations. From across the globe, questions of safety, questions of ethics, questions of why, and what comes next. The babies were named Lulu and Nana. We don't know much about them. Their real names are kept secret. Began not with a mutation, but with a choice. Someone made a decision for them, inside a lab, before their hearts began to beat. And with that act, the door to the future creaked open. You lie here now, safe in the present, but that door remains. It is neither fully closed nor fully open. It hovers. It's uncertain, waiting for us to decide how wide to let it swing. Because what happened with wondering if we can do this? Should we? And if not now, when? And if not this, then what? The edits made to Lulu and Nana were not perfect. One gene may have been partially changed. There were questions about unintended consequences, off-target effects, places in the genome. Accidentally touched CRISPR is powerful. Yes, but it is still learning, still refining, still walking the tightrope between precision and unpredictability. Yet even so, the idea remains, glowing quietly at the edge of sleep. Could we use this power to prevent suffering? Could we someday do it safely, ethically, beautifully? But, when untethered from caution, can lead us into deep waters before we've learned to swim. And so, since that day, the world has taken steps. Countries have passed laws. Commissions have formed. Scientists have spoken with greater care. Clearer voices, not to close the door completely, but to hold it with reverence. And yet, Even now, even tonight, someone testing delivery methods, watching embryos through microscopes. The science does not stop. It evolves. It listens. It waits so. We find ourselves in a moment of in-betweens. Between what has happened and what could, between fear and promise, between the first edit and the next. You lie still, listening to the hush. Perhaps in this softness, you feel the ache of curiosity, the ache of not knowing how this story ends, the sense that we've just begun a chapter we don't yet understand. But maybe that's the careful unfolding, and maybe the most human thing we can do is pause, reflect, and ask better questions. Because behind every experiment is a life, behind every gene, person, and behind every edit, a legacy we may never fully see. The twins who changed everything are growing up now, their bodies holding the shape of a decision they did not choose. We do not know who they'll become, but for what comes after. And maybe in the gentle rise of your breath, you start to wonder. A single change can echo forward through time. Can it also shape the way we age, the way we remember, the way we remain? The night deepens. You are still wrapped in warmth, floating just beyond thought. Not quite asleep, not quite awake. A gentle hush surrounds you, like velvet drawn across the world. And in that quiet, something flickers. Not light, not sound, but a question. A question that doesn't ask for urgency, only presence. Now that we can edit the script of life, should we? The tools exist, precise, powerful, astonishing. With CRISPR, we can seek out a single word in the vast book of your DNA and change it. We can correct mutations, silence genes, add new ones, even reprogram how they're read. But behind every tool is a hand, and behind every hand, a mind. And within every mind, a story, a hope, a fear, a dream. Tonight, you drift into that space, the soft, uncertain space where science meets soul. Imagine a child, not yet born. Their genome exists as potential, not yet shaped by sunlight or love, by loss or laughter. Now imagine being offered a menu, a list of possibilities, not just diseases to remove, but traits to enhance. Would you change their height, their eye color, their temperament? Would you increase their resistance to depression or give them a higher chance of musical talent? Would you want them to be stronger, faster, more focused, to carry fewer burdens than you? And if you say yes, where do you stop? This is not just a question of science. It is a question of values, of vision, of what it means to be human in a world where chance can be overwritten. Genes are not simple. They are not switches to flip. They are webs, complex, dynamic, and deeply interwoven with environment. One gene may affect many traits, and one trait may be shaped by many genes, each whispering its own instruction. To change one thread is to tug the whole fabric. Still, the dream remains. You may picture a future free of inherited disease, a future where children are born without the pain of disorders passed down through generations, cures before symptoms, health before harm. And that dream matters. It pulses with compassion, love. But intertwined with the dream are dilemmas, subtle, shifting, deeply human. If we edit out disability, do we also risk editing out diversity? If we remove traits we deem undesirable, whose definition are we using? And if we seek perfection, do we lose something sacred in the imperfections? Because genes don't just carry problems, they carry possibilities, surprises, contradictions. A gene that increases risk of one illness might also enhance creativity. A mutation that seems like a burden may hold unexpected beauty, even sorrow, shaped by certain genes. May deepen empathy or inspire art. The line between weakness and strength is not always clear, and when we edit for preference instead of need, we begin to drift, not toward healing, but toward design, toward aesthetic, toward control. Would a world filled with genetically sculpted people be more harmonious or more divided? Would choice become pressure? Would variation become error? You let these questions wash over you like waves smoothing the shore. No need to answer them all. Just let them settle, soft and curious. In many cultures, there's a belief in embracing what arrives, a reverence for what is given rather than chosen. A child is seen not as a project to perfect, but as a mystery to unfold. And yet, there is also love in wanting to spare suffering, in wanting to ease a life before it begins, in hoping to hand your child a world that is kinder, gentler, more spacious. These hopes are not wrong. They are human. But even with the power to edit, we must carry humility, because not everything in our DNA is understood. Many genes are still unnamed, their functions hazy, their effects shaped by environment, by randomness, by chance. The genome is not a finished book. It is still being written, still being read. A living manuscript, fragile and vast. So we are asked to hold two truths at once. We have the power to shape life, and we do not yet know what all the shaping means across the world. People are gathering to ask these questions together. Bioethicists, parents, doctors, philosophers, scientists, voices rising in dialogue, not to halt progress, but to guide it, to anchor it in care, in justice, respect. Because in a world where the rich might buy enhancements, where the powerful might shape generations, we must ensure that the tools of biology do not become the tools of inequality. What good is a stronger gene if it deepens a divide? So we slow down, we breathe, we listen to each other. To those who have been marginalized, excluded, silenced, to people with disabilities who live rich, full lives, to those who carry genetic risks, but also great strength. Their voices are not just valuable, they are essential. For in the end, the genome does not belong only to scientists. It belongs to all of us. It is the shared thread of humanity, delicate, mysterious, miraculous, and when we touch it, we must do so with reverence. Tonight, lie in stillness, listening not just to science, but to something older, to the pulse of wonder, to the ache of care. You feel it, don't you? This balance between possibility and responsibility, between curiosity and caution, between shaping and surrender. In this space, there are no final answers, only better questions. Should we shape intelligence? Should we erase anxiety? Should we choose boldness over gentleness, clarity over sensitivity? And who decides? You let the questions rise, unhurried, like mist above a lake. For now, it is enough to wonder. The tools are here, the dreams are vivid, the dilemmas are deep, and through them all, your breath moves slowly, a rhythm of thought, of sleep, of soul, and sleep. And, just before the drift into dream, a final question hums softly inside you. If we can shape our children's minds, might we also shape how we remember, how we grow old, how we choose to let go? You're resting now, nestled in softness, breath slow and even, outside your window. The night is quiet, and inside your body, inside every cell, a quieter world hums on, unseen but ceaseless, a world of sequences, of spirals, of signals, and you are made of it. But tonight, imagine something else, a place, a garden. It stretches wide within you, not rows of vegetables or flowers, but something wilder, a landscape woven from memory and potential, your DNA, transformed into root and leaf and winding path. There are trees, here, tall and deeply rooted, the ancient parts of you, inherited from long before you were born. There are vines twisted around forgotten memories. There are weeds, some harmless, some invasive, and there are seeds scattered in the soil, waiting for the right light to bloom. This is the garden of your genome and CRISPR. That quiet molecular tool is like a set of pruning shears, precise, sharp, intentional. It doesn't redesign the garden, but it helps us understand it. It lets us clip the diseased branches, clear the tangled paths, or encourage a new bloom to grow where before only silence lived. So you walk slowly in your imagination through this garden, and as you walk, you wonder what might be tended, what might be changed. First, you come upon a shadowed grove. Here lie the genes that carry illness, some waiting in silence, others already whispering their symptoms into the body: cystic fibrosis, Huntington's disease, sickle cell anemia. These are not just names. They are real burdens carried by real people, written into the roots of their being. Now, with CRISPR, we hold a promise to cut out the broken word, to repair the damaged sentence, to prune what once could not be touched. Imagine a child, their pain lifted before it begins. Imagine a family no longer waiting for illness to unfold. This is not fantasy. This is unfolding. Now, in clinical trials, in brave bodies, in cells returning from the lab changed and renewed. In these moments, CRISPR is a healer. The gardener's gentle hand, removing rot, clearing space for health to grow. But as you walk further, the garden changes. Now you come upon the paths of preference. Not disease, but desire. Not healing, but enhancement. Here is different, softer, perhaps, but also more uncertain. What if we edit genes to increase muscle growth, to improve memory, to enhance learning, or delay fatigue? Would we cultivate beauty, or lose the wild, surprising shapes that make us human. There are flowers here with brilliant color, but you wonder, were they planted or designed? Which blooms are worth growing? You pause beneath a flowering branch, fragrant and symmetrical. It's lovely, but so is the untamed cluster of wildflowers growing just beside it. Windblown and irregular, blooming from cracks in the path. In this garden, beauty has many forms. You begin to understand that editing isn't only about what is possible, it's about intention, about restraint, about listening to the land before deciding where to plant, because every edit is a choice, and choices echo. A single snip might prevent disease or silence something vital we don't yet understand. A well-meant change might affect not just one trait, but many. Genes are not isolated plants. They're a tangle of roots, growing together, affecting each other. That's what makes CRISPR powerful, and also delicate. You kneel now, brushing your fingers across a patch of soil. This too is part of your genome, the epigenome, the layer of life that doesn't change the DNA itself, but changes how it's read. It's like the gardener's journal, notes in the margins, guiding how each plant grows. Even here, scientists are learning to shape not only the code, but how it's expressed, like deciding not just which seeds are planted, but when to water, when to prune, when to let be. And suddenly, this garden begins to feel even more alive, because genes are not fate. They are possibility. They respond to the world, to food, to air, to emotion, to touch. Your garden blooms and bends in response to everything you live. So CRISPR is not a final answer. It is a companion, a steward, a tool that must be used with care. Because this garden is not ours alone. You'll remember now. The paths behind you stretch back generations. Your DNA carries more than traits. It carries memory, survival, long echoes of the people who came before. Their stories are etched into your cells, like tree rings, like soil layers, what they endured, what they passed down, what they preserved, all of it shaping the landscape you now walk through and forward. The path continues to children, to grandchildren, to people not yet imagined. Will we leave them a garden full of nourishment, or one so trimmed and tidy it no longer grows wild? As you near the edge of this dream space, the garden softens around. It becomes quieter, gentler. You feel its breath, not fast, not urgent, but steady, patient, because life, like a garden, moves in seasons. Not all seeds bloom at once. Not all soil needs tilling. Some things are meant to grow slow, in shadow, in mystery. CRISPR may offer sunlight. It may offer scissors, but it cannot replace the feeling of rain on leaves. The wild flight of pollen is the quiet way a seed knows when it's time to rise. And maybe that's the question, not just what we could change, but what we should let be. You linger at the edge of the garden now. You look back, taking it all in. The vibrant, the broken, the unknown, and in your chest, there is no fear, only wonder, only the soft realization that this landscape is not fixed. It is alive, and your choices, our choices, will shape what grows next. So as you rest, letting your mind return to your breath. You carry the garden with you. Somewhere in the darkness, a seed waits, and somewhere in the next chapter, a question rises with the sun. If we can cultivate life so carefully. You breathe slowly now, the night gently deepening around you. There is a stillness in the air, like the moment before a story begins, or just after a secret is whispered. In this soft space, you are not just a person. You are a bridge, a link, in a long, invisible chain, reaching backward through time and forward into futures. Yet unborn, every cell in your body holds a story, but some stories are not just yours. They belong to your ancestors. Quiet travelers passed down not by memory, but by molecule, by DNA, by the fine threads of inheritance that sewed together the fabric of generations. Tonight, we drift into that fabric. We follow the threads. We listen for echoes, because CRISPR, that careful pair of molecular scissors, does more than heal a single person. When used in the earliest stages of life, in an embryo, in an egg, in a sperm cell, it changes the very roots of a family tree. These are germline edits. They don't fade with time. They carry forward, silently, from parent to child, from child to grandchild, like ink pressed into the margins of a book, passed hand to hand. What we change today may shape someone we'll never meet. A name not yet imagined, a life not yet begun. And with that power comes a deep and delicate wonder in your mind's eye. You float through time. You see the generations unfold like ripples on water. One choice, a single tweak to a gene, becoming a wave that moves outward, touching hundreds, thousands, perhaps millions in the distant mist of tomorrow. And you ask gently, what are we leaving behind? Not just in our habits or words or stories, but in our very biology. You feel it, don't you? The quiet gravity of that idea. Not heavy, exactly, but sacred. There is a tenderness in thinking of those who come after us, and a responsibility to choose with care, to listen not only to science, but to the deep hum of humanity that runs beneath it. We have always passed things on, not just hair color or height, but resilience, vulnerability, memory. Some patterns skip a generation, some linger unseen. Folded into the body's silence, epigenetics adds another layer to this passing down. These are changes not to the genetic code itself, but to how it's read, like penciled notes in the margins of your DNA instructions that say, read this louder, or skip this part, or save for later. And these notes can be shaped by life, by stress, by love, by hunger, by trauma, by care, a mother's fear, a father's loss, a grandparent's hope. All of these can leave faint fingerprints in the biology of their descendants. Science is still learning. How? Still uncovering the ways in which emotion, environment, and experience press into our very cells, like soft impressions in clay. So even before CRISPR, we were already editing the future, not with scissors, but with living. And now, we add the possibility of precision, of intention, not just to respond to life, but to direct it, to decide what stories continue and which are gently erased. This brings comfort and also caution, because identity is not built only from strength. Sometimes It is shaped by struggle, by imperfection, by mystery. What happens when we begin to curate ourselves? When we decide what traits are worthy of being passed on and which are not? You may imagine a future child, one whose genes were edited before birth. They are healthy, brilliant, free from risk, free from pain. And yet, will they feel the weight of that design? Will they wonder who they might have been without it? Will the soft thread of their individuality feel shaped by love or expectation? We don't know. Not yet. But the questions rise like tidewater in the soul, not to frighten, only to slow us, to remind us that power must always walk with wisdom, and wisdom often grows best in quiet, in reflection, listening. You lie still now, wrapped in that quiet, and you begin to sense that your body is more than you. It is history. Alive, it is a gift passed forward, made from countless lives, shaped by time, touched by choice. Even your mitochondrial DNA, the tiny loop of genes in your cell's powerhouses, comes only from your mother, and her mother, and her mother. Before that, like a candle flame, carried down a line of women, across centuries, the choices they made, the foods they ate, the stress they carried, all of it echoing in you as softly as breath. And now, for the first time in human history, we hold the tools to shape what echoes next, to edit not only what is broken, but what is becoming. You begin to feel the edges of this truth, that we are not just editing genes. We are editing futures, that sterile rooms and glowing screens hold choices meant for people not yet born, that the silence after an edit is not empty. It is filled with potential, a whisper that will one day become a voice. So we must ask, who do we edit for? What values do we carry into the lab? Do we make choices only for health, or also for perfection? Do we erase risk, or erase difference? And who gets to decide? These questions do not ask for answers tonight. Only your attention, only your heart. You drift now back through the threads, through the garden, through the spiral code. You return to your breath, to your body, knowing that you, too, are an echo and a beginning, and somewhere not far from this moment. Scientists are learning how to repair memory, how to protect the brain from the fading light of time, how to hold on to the self when it begins to unravel. Because if we can shape what we pass on, might we also learn to hold on to what we forget? The air is soft tonight. Your body is still, held by sleep's slow rhythm, but your mind wanders, not anxiously, not far, just gently, like a leaf drifting downstream. You float through thought, through memory, through science and story, and you return again to CRISPR, that elegant tool, that silent scalpel, the editor of life's long code. But tonight, we don't celebrate its precision. We sit quietly with its limits. We watch the places where certainty, FRAY, where even the cleanest cuts can leave something jagged behind. Because even light casts a shadow. Even clarity has edges, and no tool, matter how advanced, is perfect. CRISPR was designed to be exact, a molecular guide, leading enzymes to just the right spot in the DNA, like a hiker using a compass to find a hidden clearing in a vast, tangled forest. But sometimes, Even with the best directions, the trail forks, the wind shifts, the enzyme lands in the wrong place and makes a cut it was never meant to make. These are called off-target effects. Edits made, not to the gene in question, but to another, unrelated, unexpected. It's as if you meant to prune a single rose and accidentally clipped the vine beside it sometimes. The result is harmless, a change too small to matter, a note barely out of tune, but sometimes the consequences echo. They may not appear for years, or not until the edit is passed to the next generation. And that's part of the mystery. Even with all we know, the genome is still a jungle, layered, interconnected, full of hidden paths. We don't yet understand it all, and what we don't understand can't fully control, so we tread carefully. We pause between each breath. We let uncertainty sit beside us, not as a threat, but as a reminder that we are still learning, still listening, still in the early chapters of a very long book. You close your eyes and see a single cell, newly edited, its DNA adjusted, its path redirected. But cells divide. They copy themselves. They replicate. They grow. And sometimes, not every copy carries the same change. This is called meiosis. It means some cells in a person carry the edit and some do not. A body built like a patchwork quilt. Some squares altered, some untouched. A quiet complexity, invisible on the surface. And this, too, brings questions. Will the edit be strong enough to matter? Will the unedited cells work against it? How do we measure success when the answer lives beneath the skin, layered in silence? There are other limits, two subtler ones. The cell's repair system doesn't always behave as we expect. Sometimes, after CRISPR makes its cut, the DNA repairs itself imperfectly. It may insert or delete pieces, creating outcomes we didn't plan. It's like trying to edit a poem and discovering that the page rewrote itself while you weren't looking. These outcomes, known as indels, are not necessarily harmful, but they are unpredictable. And unpredictability, in a system as delicate as life, requires reverence. So, tonight, we lie with this truth. CRISPR is brilliant. CRISPR is beautiful. And CRISPR is not flawless. Its precision is powerful, but its edges are still soft, still learning, still being shaped. By trial, by error, by wonder. And that's not failure. It's the nature of science. Because science is not a straight line. It is a spiral. A spiral, a spiral inside a spiral. Just like the DNA it seeks to understand. We move forward. Yes, but also around. We revisit, revise, return, and each time we learn a little more in your stillness. This feels comforting because you, too, are not perfect. You, too, are made of revisions, of repairs that didn't quite mend the first time, of moments that branched unexpectedly and became something new. You are not a flawless story. You are a living one. And so is CRISPR, not a magic wand, but a companion, inviting us to listen more clausolito, the language of life. There's a strange beauty in imperfection, in letting go of total control, in understanding that mystery is not the opposite of truth, but a partner to it. We cannot eliminate all risk. We cannot promise that every edit will go as planned, but we can hold these truths gently. We can ask better questions. We can slow down when it matters most, and sometimes slowing down is the most radical act of care. You picture a lab now, dimly lit. A scientist leans over a microscope, not with haste, but with awe. There's no rush in her movements, only curiosity, only listening. Because when working at the edge of life, listening is more important than knowing. The genome does not shout, it whispers, and if we are to guide it, to shape it, we must whisper back, not with certainty, but with humility. Tonight, you carry that with you, not fear, but a softness, a respect for the things that don't fit neatly into models, for the outcomes we didn't expect, for the truths still forming. Even the sharpest tool can slip. Even the clearest map can mislead. Even the brightest light can cast strange shadows. And yet, we go on. Because even in its limits, CRISPR reveals something vast. It reminds us how complex life is, how layered, how wondrous, and how much of it still waits to be understood. So, as you drift now, held by darkness, you do not seek perfection. You seek presence. You seek care. You seek the courage to hold precision and uncertainty in the same hand. And somewhere, far ahead in the folds of your brain, in the edges of your memory, another question is forming. If edits can miss their mark in the body, what happens when we try to edit the mind? You rest beneath the ceiling of stars, eyes closed, yet open to the hush of the world. Your breath slows, soft as dusk, and something deeper begins to stir, not inside you, but around you. A murmur from the soil, a whisper through branches, a rhythm in the wings of things unseen. Life does not speak with words, but with signals. And CRISPR, that silent editor of life, listens in tonight. You leave the boundary of your own skin. You drift outward, past veins and thoughts and memories, into the greater body that holds us all, the body of Earth. The planet pulses not just with weather and water, but with genes, billions upon billions of instructions coded in the living things that walk, swim, bloom, or hover. And in this vast, breathing system, CRISPR is more than medicine. It becomes ecology. It becomes agriculture. It becomes a new kind of stewardship. You float over a field now, golden, rippling, quiet. These are not wild grasses, but crops shaped by centuries of human care. Wheat, corn, rice, soy, each with a history braided into your own. Each nourished by sun, rain, and the slow, invisible force of inheritance. But crops face challenges. Frost that comes too early. Pests that nibbled through leaf and root. Droughts that stretch the soil thin. And diseases that arrive without warning, devouring entire harvests before they reach your plate. Scientists are using CRISPR to make these plants more resilient, not by adding foreign genes, but by unlocking possibilities already hidden within their DNA. A tomato that ripens more slowly keeping its sweetness longer. A banana resistant to a blight that once threatened to erase it. Wheat that bends less easily to fungus. Rice that yields more grain in less land. These are small changes. A silence there, but they ripple outward, changing what grows and what survives. And it is not only crops beneath your drifting mind. A mosquito hums, tiny, persistent, often deadly. Mosquitoes carry diseases that kill hundreds of thousands each year. Malaria, dengue, Zika, yellow fever. But what if they didn't? What if, with CRISPR, we could render a mosquito unable to spread infection? What if we could reduce their populations by editing a single gene that controls fertility? This is more than an idea. Gene drives a form of CRISPR that ensures a genetic change spreads through a population rapidly are already being tested. Imagine a world without malaria, without the tiny bite that brings fever and loss. But as you float deeper into this idea, you also feel the tension beneath it. What else might change if mosquitoes disappeared? Birds eat them. Fish feed on their larvae. Predators depend on their buzzing presence. What seems like a simple edit echoes through the web of life, and here, again, you find yourself in the quiet space between what can be done and what should be. CRISPR's promise is not limited to survival. It extends now to resurrection. Species once lost, the passenger pigeon, the woolly mammoth, the northern white rhino, are being considered for genetic revival through ancient DNA, through closely related living species. Scientists imagine bringing back what was lost, extinction. They call it a word that sounds almost like hope, or perhaps like reckoning. But would these creatures still belong? Would they thrive in a world changed so far from the one they knew? If we bring them back, are we healing the past or rewriting it? You feel it now, the thread that runs through every intention. CRISPR is not a wild wind. It is a tool in human hands, and those hands carry the weight of vision, of caution, of humility. Because when we change a plant, an insect, an animal, we are not just changing them. We are shifting balances, redrawing boundaries, sculpting ecosystems that evolved over millions of years. Still, there are places where CRISPR may bring balance back. In the oceans, coral reefs bleach and crumble under warming seas. But what if we could give corals a better chance, a tweak to tolerate higher temperatures, a change to strengthen their partnership with algae? Could gene editing help a reef survive where otherwise it would vanish? Could it offer resilience, not as domination, but as collaboration? These questions do not have easy answers, but they rise like tidewater all around us. CRISPR is not inherently good or bad. It is possibility. It is a sentence unfinished. You may wonder, where does the blueprint end? Does every living thing have a right to remain untouched? Or are we part of the system we seek to tend, not above it, but within it? You feel this deeply, lying still in the hush of night, the sense that life is not made of walls and edges, but of gradients, of connections. The gene of a flower may echo in the gene of a fish. The survival of one species may depend on the silence of another, and in our urge to help, we must not forget to listen. CRISPR's reach extends, not with speed, but with attention, with the slow, deliberate unfolding of a science that dares to look closely, to question softly, to imagine what life could become if guided with care. You breathe in now, deep and slow. You picture the planet not as a sphere, but as a living manuscript. Its language not written in ink, but in base pairs, in feathers, in roots, in hooves, in wings, and CRISPR. CRISPR is a pen held lightly, not to overwrite the story, but perhaps to repair a sentence, to restore a voice, to adjust a rhythm that has faltered. You don't need all the answers tonight, only the awareness that we live in, relationship, that every change to a single cell can echo into the forest, into the sky, into the soft turning of the earth beneath us. And as your thoughts quiet, you feel one more question rise, not loud, but steady. If we can shape the genes of the world around us, what happens when we turn inward again and try to rewrite the very clock of life? Tonight, the world is hushed. No more questions press urgently. No machinery hums. No microscopes gleam under the hum of fluorescent light. Tonight, there is only you and the quiet rhythm of breath, a body at rest, a mind floating, drifting across the edge of wonder. You have traveled a long road through the folds of your DNA, into the garden of your cells. Past the sharp edge of possibility, you have seen what CRISPR can do. You have imagined what it might still become. And now you pause. In that stillness, one question lingers. If we can change so much, so precisely, so powerfully, who do we become? Do we shed our frailty and become something more refined? Do we hold on to our flaws as if they are essential? Or do we gently edit them away? Do we aim for perfection or discover that perfection was never the point? You lie quietly. The question doesn't need to be answered. Not now. Not yet. It only wants to be heard. Because this isn't just about genes. It never was. It's about what it means to be human. When the story of being human is no longer fixed, when the ink is still wet and the pen is in our hand, you picture yourself not as one person, but as one page in a great unfolding manuscript, a living text, written in laughter, in failure, in memories, in mistakes, in hands that held, in hearts that broke. In the delicate friction of trying, we are not only our DNA, we are what we've built on top of it. Kindness is not genetic. Empathy is not edited into us. It is grown, cultivated in the quiet dark of experience, in the long, slow dance of being with others. So what happens, you wonder, when we begin to change the foundation, when we redesign, not to heal, but to enhance, a sharper mind, a stronger body, a longer life? Will we use it to lift each other up, or to rise alone? CRISPR offers no morality. It offers questions, and the answers are not in the petri dish. They are in us, in the soft, fragile space where science meets soul. You think of the children yet to be born, children shaped, perhaps by intention, children without certain risks, without certain traits, children who may never know what it means to be less than. But you also wonder, Will they know what it means to be enough? You drift deeper now, into a world that flickers like starlight, not fully formed. You're not fully distant. A world in which CRISPR has become as common as vaccines, as expected as electricity, where illness fades and aging softens. A world where parents edit not just for survival, but for advantage, for beauty, for brilliance, for control. Would this future be better, safer, fairer, or just ... more? More of everything. More choice, more pressure, more distance from what was once natural. You breathe in slowly. Wind, not clinging, not deciding, just listening, because that's what we must do now. We must listen, not only to the science, but to each other, to those who carry old wounds, to those who live at the edges of what society accepts, to those whose voices were long ignored, but whose lives may be altered most. The question isn't just what can we change? It's what must we protect? Compassion, curiosity, diversity, a sense of wonder, and the strange imperfect joy of being human. You think of the first time a child laughs, of how memory clings to scent, of the way sorrow softens the heart's edges, and how loss teaches you to love harder. Would we edit those lessons out? Would we trade pain for predictability? Or is there something essential in struggle, not for its own sake, but for how it shapes the light that comes after? You feel this now in your chest, a quiet ache of not knowing, and still, it comforts you, because mystery is not failure. It is invitation, an open hand held out to tomorrow. You are a part of that tomorrow, whether you choose to edit or not, whether you change the code or simply live within it. You are already changing the world through your questions, through your presence, through the softness with which you hold complexity in this future. The one unfolding at the edge of midnight needs softness. It needs dreamers and doubters and people who pause before they decide. People like you. You shift slightly beneath your blanket, the weight of the day releasing its final hold in this near sleep. You imagine the next generation of scientists, not rushing toward certainty, but walking gently through ambiguity, not seeing the genome as a machine to be repaired, but as a garden, wild, layered, and alive. They will make mistakes, just as we have. They will ask questions that unsettle. They will stumble, but maybe, with time, they'll also find a deeper kind of wisdom, not the kind that edits perfectly, but the kind that edits with love, because that, in the end, is the real frontier. Not what we can change, but how we hold what we change. Do we wield this power like a sword or like a lullaby? Do we edit to escape being human or to better understand what it means? You don't need to know, not tonight. You only need to rest, knowing that the story continues not as a line, but as a spiral written in stars and cells, in silences and symphonies. In moments just like this one, you breathe in once more, the air cool and gentle. You are not the end of the story. You are a turning point, a listener, a question, a light still becoming. And as you drift into sleep, you wonder, soft and slow, If we are learning how to change everything, what will we choose to cherish?